Victoria, you want to go ahead?
Yeah, thanks, Greg. So welcome everyone to the first SOCAAR Seminar of 2021. My name is Victoria Arundel. I'm in the Dalalana School of Public Health, but I'm very pleased to introduce today's speaker, Dr. Chris Carlsten. Dr. Chris Carlsten is a professor of medicine and head of respiratory medicine in the Faculty of Medicine at the University of British Columbia. He is a leading clinician scientist, and he holds both the Canada Research Chair and the AstraZeneca Chair in Occupational and Environmental Lung Disease. He's also the director of the Air Pollution Exposure Laboratory, which is probably how most of you know him. His lab is highly collaborative, it's interdisciplinary, and it's very productive, making important scientific contributions to our understanding of occupational and environmental exposures and disease. Their research focuses on respiratory and immunological health effects of inhaled environmental and occupational exposures like diesel engine exhaust, western red cedar, and phthalates as their model exposures. They use both human exposure studies as well as genomics to investigate the synergies between co-exposures, because most of us are not exposed to single things, and how these co-exposures mediate health effects related to airways disease. And all of this work dovetails very nicely with Chris's clinical training and expertise in internal, occupational, pulmonary, and critical care medicine. And today, Chris is going to talk to us about using controlled human experiments to better understand the health effects of reducing the particulate component of diesel engine exhaust in the context of traffic-related air pollution. And this is a topic of great interest in the workplace where I focus, as well as the community, as diesel engine technologies continue to change and hopefully improve over time. So welcome, Chris, and thank you for speaking with us today.
Wow. Honestly, I think that's the nicest introduction I've ever got. I'm not kidding about that. Thank you. Really. So, and thanks, Min, for saving me once again. Min regularly bails me out of technological challenges. So thanks, Min. Yeah, you can go ahead, please, on the next one. Well, this part I'm very comfortable talking about, while at the same time speaking to a audience which is enriched with aerosol specialists, including chemists, etc. Makes me a little bit nervous about the technical aspects of particle filtration, which we'll get to. But I'll cry uncle on that if necessary. But again, as a high level statement, I'm very comfortable saying that pollution is a massive problem in my kind of mid-career reflections. It hasn't, unfortunately, gotten all that much better since I was in early training. It kind of has, it kind of hasn't. Some places have got cleaner, some not. But the overall burden, whether we're documenting it better or not, it just seems more and more devastating. So 9 million annual deaths is a huge number every year, obviously. I mean, I try not to say the following, but I can't help but say it's a lot more still than COVID, although obviously COVID is a devastating problem and may or may not interact significantly with air pollution. I'm not going to talk more about COVID, however. Go ahead, please. So, and just scroll a couple more so I can save time. And yeah, that's good. So the overall framework for our lab, and Victoria introduced it really well, is that not only co-exposures, which we'll get to, but to really understand these complex issues of exposure science and how they affect policy and real-world application. I use the word plausibility a lot, and for me, plausibility requires interdisciplinary studies, whether it's toxicology, epidemiology, or what we specialize in, controlled human exposures. And we do some cellular work, we do some epidemiology, but really we've come to focus on controlled human exposure studies as something we think we can be an example for others. Go ahead, man. It looks, like this, if you like a visual, something that we kind of articulated in this paper, but it's really the same concept that I said before. Go ahead, man. And specifically in our lab, we won't go through every one of these pieces, but just to give you a sense of how I and my team kind of envision the pieces with impact being ultimately what we hope to have. Getting from the inputs to the impact is not easy, but we're increasingly trying to embrace that challenge, and as Victoria said, doing it in a collaborative way. But whatever exposures, the ones you see are the ones we focus on most, can lead to outputs which can be as simple as symptom data or as complex as omic data, including DNA methylation, etc. I'll just show you a little bit of omic data in today's presentation, but it's something we've done significantly in working with, whether it's a regulatory agency, a compensation board like WorkSafe BC, etc. We've chipped away at these relationships to make impact, but we have a very long way to go. Go ahead, Min. And just to further kind of make it tangible and slightly more granular and visual, and some of you have seen this, but anyone's welcome to once travel is a happy thing again. But basically, the whole system together works by recruiting humans with our team. That's very good at that not easy task. Getting them to be exposed in a safe way, I'll talk a little bit more about. That's number two. Oftentimes doing bronchoscopy to get samples from the lungs, and then bringing those samples back to a lab where we can analyze them, and all that together helps us with the picture I'm going to try to paint a bit more now. Go ahead, Min. So this is what it looks like a little closer up in the lab. Many of you will be familiar with these equipment that are not quite as new anymore as when I started, but still work very well. In fact, some of them have been replaced since this old picture. And as Victoria was saying, we look at multiple exposures, and the other thing that we do that's not unique, but something I want to point out because it's critical to every one of our results, is a crossover design. So that means that every individual, like this one you see in the picture, for example, will get every exposure that's in whatever experiment we happen to be doing. So the only thing that changes, and we work really, really hard to make that true, the only thing that changes is the exposure. We do whatever we can to keep everything else identical, so that when we see results, we can say with good confidence that that is because of the exposure that was changed. Now, you can't say that 100%, but it's about as good as it gets. It's like, or it is not, it's not like, but it is a randomized controlled trial, and it's just as much of a randomized controlled trial as a drug study that, you know, gets a drug approved for bajillion-dollar pharma initiative. So it's the same design that's held to that level of standard from Health Canada, from the NIH and the FDA in the US, etc. Go ahead, Min. These exposures are equivalent roughly to this kind of day in Tiananmen Square, for example, but unfortunately, even equivalent to summertime exposures that we've had here with wood smoke. And it's not in the previous slide, but we actually can expose to wood smoke with a new system that we've added to our lab. So this same picture you see here with me and this research participant can be used for wood smoke exposures now. It's kind of a modular design that we're happy to have now. So we're really approximating very much whether it's Asian exposures, whether it's occupational exposures that occur even in Canada, or whether it's wood smoke exposures that occur practically every summer in BC now. Go ahead, Min. For those that wonder about the ethics of this, that's a whole nother lecture, but this is one paper, and there are others that I can refer you to from independent experts that vouch for the importance of this under safe circumstances such as ours. Go ahead, Min. And again, I'm not going to go through all these, but if you really want to think more about the issue of co-exposures and how important they are, you can look at this review paper. The highlighted oval is the combination of diesel exhaust or diesel exhaust particulate, DEP, and allergen. And really, I started here at UBC with that combination in mind, inspired by some studies in Southern California, and that's been a huge part of our work. Go ahead, Min. And this is how we conceptualize it: allergen and diesel exhaust, and then trying to fill in this black box, focusing on airways disease. But some of the endpoints that we've looked at from an inflammatory perspective are also important to cardiovascular disease. The GXE, or G by E, in the middle is gene-environment interactions. So all of the endpoints that we look at, practically speaking, we look to see if there's a component of the individual's genetics that modifies these relationships. Next slide. And again, not going through these, but these are the sort of a sampling, not particularly updated apparently, of the kinds of findings that we've published to understand this relationship, and it all adds to that plausibility. And I think I'll say this later again, but just as a interesting preview, I didn't realize when I started doing this that the whole green environment or greenness science would become such a big thing, but this has now become really important. And again, it's part of the interesting sort of serendipity of science that with the study of greenness, one of the outstanding questions remains, when you bring greenness in proximity to traffic, the greenness seems to be generally a health promoting factor, but when it's next to traffic, it may not be quite such a good thing because of these biological relationships that we've been describing. Go ahead, man. You can, keep going to the next one. So for this talk, I know there's a bit of a long introduction, but I really want people that aren't familiar, I need them to understand the background. We wanted to look at diesel exhaust, which is particles and gases. And then if you go to the next slide, Min, we want to remove the particles. That's good. Okay, you can keep going. Remove the particles and see how this relates to previous work we had done. So in this slide here, before we got into the particle depletion, we did just the combination of diesel exhaust and allergen, and it's all done by bronchoscopy. One of the techniques that sort of took a while to establish and sort of in the rear view mirror now, but these were very difficult things early on. And we were able to show the various inflammatory phenomena that you see here, enhanced by the combination of diesel and allergen, or DEA. Go ahead, Min. And this is just so you see like the real deal, so to speak. The red oval is the fluid from the lung that results from the combination of diesel and allergen. And if you even just eyeball it and you're not familiar with this, you can kind of see it's a thicker, more cellular, more opaque fluid. It has more eosinophils, a lot more eosinophils. And we get other samples through brushing and biopsy. We're not going to talk about all of these, but just to give you a sense of what we do. Go ahead, man. Okay, so now coming to this design of the particle depletion study. I know this is the reason I wanted to set this up so much is so that this wouldn't be hopefully so confusing. But again, crossover study. So each of the bars, gray, yellow, blue, red, are one of the crossover arms. So every individual gets each one of those randomized to order. So the order is different for every person, which is an important part of the validity of the design. Get bronchoscopy, as I said. But the difference here is the red PDDE, and that stands for particle depleted diesel exhaust. So that's the new thing we did, at least in terms of the context of combined allergen exposure. And right around this time was Dieselgate. And people will remember, I'm not going to go through the whole history, but it was coincidental that this happened and that we were made aware that on-road testing of NOx was much, much higher, up to 40 times higher than what the companies, not only Volkswagen, but others had reported from their very kind of perfect scenario in the lab. Go ahead, man. And this is where, like I said, I'm happy to learn from you guys, because it's really not my expertise how these technologies work in any detail. But the filters themselves, now, diesel engine technology is more complicated than just filters. But if we just think about the filters that are designed to remove the particulate matter, they do a great job at that. But much more than I even realized when I started with this, they increase NO2 levels in particular, and sometimes NOx in general. And if you don't have additional technology to control those gases, then there may be a trade-off that you may or may not be happy with as you see the results. But the real-world implications of that, the fact that NO2 or NOx could be increased was well known, as I found kind of in retrospect. But what that does to health effects is really not known, and still there are not a lot of studies of this beyond ours. Go ahead, please. So what we showed, just to be clear that this is in fact what is happening, is that the particulate matter was decreased by 94%. You can see that in the first red box. But the NO2 level went up considerably, while volatile organics didn't change a whole ton between the particle depleted and the whole diesel exhaust. Go ahead. And this slide is really about the acute effects. And we found, if you look at the red line, that this particle depletion would drop airflow. FEV1 is a measure of airflow, so it's the first second of a forced breath. And when we measure that, it's always expected that airflow will drop within 30 minutes or an hour with allergen, which is the A in this diagram. And that's, if you don't see that, then there's something wrong with your study, because allergen in these individuals who are sensitive to allergen should drop airflow just like this. Even if they don't feel it, you can measure it in this kind of 15-ish, 10 to 15% drop. But with the particle depletion, the bottom line in red, you can see a greater drop right there. And that was statistically significant. And then it all kind of came together and started back towards baseline. And by 2 days, it was. And that's an important thing, because, first of all, the individuals don't feel this. They don't complain about it per se. We measure it. And then we continue to measure it. And then by the end of the study, they're back to baseline. Go ahead. Oh, sorry. This is important, that we, as I said before, we look at genetics as a modifying factor, and we specifically look at oxidative stress, a set of genes that mediate oxidative stress. And what we did new in this study is, rather than looking at one particular gene, we've been accustomed to looking at a given glutathione as transferase gene. There's several in the family, but for example, GSTP1 or GSTM1. But we looked at a whole suite of not only the GSTs, but others that mediate oxidative stress to see whether that in fact changed this relationship, and it did. So those that had more variants, which is the equivalent of having less oxidative stress defense, because the variants will produce less of the protein that help us with protecting against oxidative stress, those individuals had a larger decline in their FEV1. So you see in the oval on the left around the red, the point at the bottom of the red line, you see that there's variability there, and indeed there is. That's the real world. That's what humans do. They vary. But within that, on an individual basis, those that had the oxidative stress more prone to damage per se were the ones that had the greatest change with the particle depletion. Go ahead. Now, this was unexpected, and I'm going to explain this because I think it's potentially important. The darker purple or mauve, if you will, are those that are atopic. All of the individuals in this study are atopic, meaning that they're sensitive to allergen, and then we use that allergen to provoke them on purpose, as I described before. But the ones in the darker color don't have pre-existing airway hyperreactivity, AHR. And AHR means that if you stimulate the lungs with a non-specific stimulus, they'll contract. And that's how we actually detect asthma. That's how we, in the real world, the real clinical world, prove that someone has asthma, is we purposely try to trigger their airways in a very controlled, safe fashion, done all around the world essentially the same way. to prove that that's real and it's not just someone's sense that they get tight when they're exposed to whatever bothers them. But the darker color are those that didn't have that phenomenon, and yet they're the ones that with this study of these exposures had the biggest change. So if you came in, it was counterintuitive, frankly. If you came in with asthma or AHR, you had some effect, but not nearly as much as those that didn't. Now, again, this all reverses back to baseline, so there's no harm done. But if this is repeated in China or in India or even here with 3 months of summertime fire smoke, this could become a permanent phenomenon in the real world, and that's the implication of this that concerns us. Go ahead. I'll probably go a little quicker through some of these additional results, but it's important not only to look at the lung, but to look at the systemic inflammation, meaning the blood, what's circulating. And when we look there, as opposed to those very acute airflow drops, there was an effect, but across the board of all the exposures, whether it was just the allergen, diesel and allergen, et cetera. There are different patterns here that you can see, whether you're looking at eosinophils, et cetera. There's an example, for example, with the 24-hour neutrophils, that with the particle depleted exhaust, that the effect was not statistically significant. But in general, in the systemic inflammation, 1 to 2 days out, the difference that we saw in the airflow was not really there, which makes sense because that difference, which is probably due to the nitrogen dioxide or NO2 that I showed you earlier, is not likely to have an effect in the bloodstream. Go ahead. And this was corroborated in a sense with this measure of nitrosative stress, which is a kind of cousin to oxidative stress, looking in the urine at 3-nitrotyrosine. And again, all of the exposures increased that marker, but it wasn't really particular to the particle depletion. Go ahead. And then Min, who's not only a IT wizard, but much more importantly, really a excellent scientist who's finishing his PhD, showed here in this paper from Thorax that not all is kind of lost, or we don't need to kind of throw these filters into the waste bin because of the nitrogen dioxide factor, because it's much more complicated than that. And there are markers like these that actually were enhanced by particle depletion, and these are good markers. If we don't have time, but if we briefly read the details at the bottom, or if you do that at your leisure, these are markers that actually are helpful for homeostasis. And so on these in these examples, the particle depletion actually helped. And so maybe they're mediated by the particles. That would be the hypothesis. Go ahead, man. And that fits also with the latest data that we have at 48 hours in the lungs, that here again, there's not such a difference between the particle depletion and the other exposures. Again, because by 48 hours, that gaseous effect is much less likely to be persistent, as opposed to particle-driven effects, which because of the way that particles stay in the airways for longer than gases do, is probably, or at least hypothetically, an explanation for this pattern. Go ahead. So the summary of that so far, and that's mostly the work from Denise Wooding, who's now in medical school here, and Min, as I mentioned, is that acutely, as I said, probably because of the increased nitrogen dioxide, there was a drop in airflow. And it was enhanced by those susceptible to oxidative stress by virtue of that gene score. And it was worse in those that didn't have pre-existing airway responsiveness, leading to that concern for what that could mean for those individuals if it was a long-term rather than just a short-term experimental study. Systemic inflammation was more similar between the various co-exposures. there was a sense of or a hint or a pattern that's consistent with homeostasis in some of the markers with the particle depletion that was in Min's study. But if you look at it overall, there certainly was not a protective effect across the board of particle depletion. And for me, that was enough to sort of question at least the assumption that just removing particles was going to for sure be a good thing for health. It may be, it may not, but it's certainly not a slam dunk pattern. So we wanted to go further and look at transcriptomics, which is the next section. Go ahead, Min. And this is Lenneth Lee, who's a master's student in the lab. You may or may not be familiar with RNA sequencing, but it's sort of the latest iteration of further and further detail into looking at the gene expression. In this case, rather than just picking certain genes and putting them on a panel, be they thousands or tens of thousands, this looks at all the genes that the technology can cover, which is the vast majority. So adds detail, not just looking at what we thought was important, but looking at all of the genes that current technology can detect in terms of their expression. Go ahead, Min. This is the pipeline. There are probably at least a few people in the audience that think about these things, but long story short is that we went to the best place we thought was available, Genome Quebec, and got the high quality data from them. Of course, the quality of the samples was important, and we tried to ensure that by the design of the study and my lab being attentive to detail. But once we get the data from Genome Quebec, using various informatics programs, including the one you see, the playfully named salmon, etc., to get the final data that I'll show you in the next few slides. So Venn diagrams, they're hopefully intuitive, and hopefully you're a bit tuned into the acronyms. So the diesel exhaust with allergen at the top in blue and the particle depleted in red. But basically what you see is that whatever co-exposure we look at, there's a unique pattern, but there's also some overlap. And hopefully that's intuitive from the nature of these Venn diagrams. You also see that it's not always genes that have increased expression. Sometimes their expression is decreased. And while we won't talk about DNA methylation now, that shouldn't be so surprising given what we know about DNA methylation and how air pollution can affect it, leading to sometimes increased expression and sometimes decreased. Go ahead. But for more insight, what Lenneth did appropriately is look at these differences at more detail, and one way to do that is in volcano plots. And the volcano plots do several things, but the important cutoffs really regard significance, which is corrected for all the multiple comparisons, and then the magnitude of the change. So on the bottom, the fold change is magnitude, and on the left is the significance. And in the corners in red, you have changes that are both very significant, even after correcting for the comparisons, but also have a relatively large change so that biologically they're more likely to actually be meaningful. And I won't go into the exact examples except for a few that I'll show you, but this is just to give you a snapshot. Go ahead. So again, the kind of thing that's not for picking through each exact piece, but that data that I showed you previously will go into these kinds of, or we'll go into a software package that will generate these kinds of maps that can show you nodes and show you the pathways that are most significantly changed by these different exposures. And it's hard to see, but the one with the arrow that is in green is cellular responses to cytokines or chemokines. And chemokines specifically are proteins that attract important cells, whether they are neutrophils or others. And I'll show you a little bit more about that. But these are the circulating proteins that are basically signaling to the key white blood cells and other cells in our body to either migrate or have other functions. So here's some more detail about that. One of those white blood cells are lymphocytes, and lymphocytes include the specific subtype of lymphocytes known as B lymphocytes, which are important because they generate antibodies. So CXCL13 is an attractant for that, and you can see that the particle depletion actually decreased that. If you look at the box furthest to the left and the two rightmost box plots of that segment, and then eosinophils, which are important to asthma and allergic biology in the middle, you can see, and then an example of several metalloproteinases, in this case MMP12, which are well known, especially in COPD, but in airways in general, to be critical. If they're not controlled, they can lead to destruction of the tissue. So in this case, perhaps particle depletion was in fact beneficial because, as you can see, it decreased the level of this MMP12. Go ahead. So with that, we added the layer of RNA sequencing to give a bit more detail and add to this conclusion, or at least tentative conclusion that I have, that it's one thing to design these technologies, but when they're actually employed and they're tested in a controlled setting with humans, we find that they may not be as simple a remedy for complex aerosol health effects as one might hope. There is, as I showed, some benefits that may outweigh the negative effects of the gases. And again, with the proper gas controlling technology, you may have the best of both worlds. Co-exposures continue to be shown to be critically important. It's almost impossible to show that co-exposures are not different. The range of genomic and other endpoints we've looked at, I gave you a snapshot, tend to always show that without appreciating those co-exposures, you're missing a lot of the detail that's probably important to the biology. The genetics I talked about, and I said this at the outset in terms of having impact and translation, I want these to become important to regulatory agencies. And I look to all of you and others that I'm working with to try to understand how to communicate this and translate this so that it becomes part of implementation. And as I said, that's kind of the next phase of our work, and it's probably the hardest, and we're working hard in that regard. Go ahead, and. You can skip over this. It's just it's basically that there are some other people that have kind of recognized this and think it's important and tied some of the pieces together that I didn't even do myself. They understood kind of the implications probably better than me, so I can share these references with you if you want. I mentioned this before. The issue of biological plausibility. I mentioned the top middle about greenness and how much variability there is in that literature, and I think this is part of the reason. Reminding you that even in Vancouver, we have events like in the upper left with these kinds of exposures. So this is not just a fantasy or something that only happens in Asia. Again, interdisciplinary work is absolutely critical. Go ahead, man. And finally, I thought I had fixed this. I pointed out Susie for another talk, but Susie's great, but not the focus of this one. I would have moved the arrow to Min if I could, but wanted to thank collaborators, too many to list, and funding agencies, and hope that I left enough time for some questions, because I really am very open to all kinds of feedback, including critical feedback, because I know that the aerosol science is not something that I understand as much as many of the people in this audience. So thank you.
Well, thank you, Chris, for a wonderful talk. Question, Jeff, you got a question?
Well, I was going to say thanks, Chris. I could think of a question. So just you pointed right to NO2. Lots of questions, but you pointed right to NO2 when you thought about still seeing responses when the particles were removed. But what about other gases? There's VOCs in there of different characteristics. What makes you think it's the NO2?
Yeah. Good question, Jeff, and thanks for coming. So the VOCs I did show, they didn't change. They weren't changed. I mean, maybe specific VOCs. We didn't delineate particular VOCs, but VOCs as a whole, total VOCs, were not changed by the particle depletion. If you had a specific, yeah, thanks, man. If you had a specific candidate, we might look into that, but I don't have one that I think could be nearly as good of a candidate as NO2. The NO2, getting back to that. Well, we know that NO2 is a critical gas amongst the many gases in complex traffic-related air pollution. But not only that, the literature on NO2 and airway inflammation and the endpoints that I've shown or highlighted long predates this. So it fits really well into the pre-existing literature. I mean, things like sulfates, we don't have a lot of because of the fuel we use. So I didn't so much suspect that. But yeah, I'd be definitely open to particular candidates that you think we should try and test.
And just remind me, this is where the allergens were instilled in one lung and not the other. So it's not a co-inhalation of allergens; it's allergens instilled. Is that right?
So yes, for yeah, I was just thinking because we've done a few of them, including some that are actually inhaled. But yes, for this study, it was instilled.
Okay.
Unless somebody like Min's going to correct me, it's always embarrassing if I misremember that particular thing. But I think that's true.
And in the inhaled model, it's inhaled first, and then the diesel exhaust are not inhaled at the same time. Is that right?
Diesel exhaust is inhaled first because while they both can lead to some airway constriction, allergen is going to lead to more of it. And if you have major airway constriction, then the particles aren't going to get in the way that they would naturally with natural airflow. So yeah, it's diesel first and then allergen second.
Okay, and just one last question. Then why did NO2 go up so much here?
Say that again.
So why did NO2 go up?
Well, I mean, that's again something you guys can tell me, but it's well described with these particulate filters if they don't have selective catalytic reduction or some other urea base system. And in our hands, it may be that part of the particle depletion uses an electrostatic precipitator. And that's a limitation of our study, which we recognize in our papers, that electrostatic precipitation, while it's important to kind of home filtration environments and other industrial environments, is not the typical way to remove particles from a vehicle engine. But nonetheless, our model leads to higher NO2, which is seen with vehicles that use only the filtration without the gaseous removal, particularly if they've been manipulated in the ways that we know that engines are.
Well, presumably NOx stayed the same, but you tipped the balance from NO to NO2. Is that what you mean? What happened here?
That's right. That's right.
All right.
Are there other questions? I've got lots, but I'd like to let others have time for questions. And please identify yourself when you're asking the question.
Hi, it's Victoria. Thanks, Chris. That was really interesting. My question's a little bit tangential maybe, but what happens to the PAHs when you remove the particles?
I don't think we know. I've done a bit of work with Arthur, but not on this particular study. So I'm just trying to think back if we've ever looked specifically at PAHs. I don't think we have, and anybody from my lab that's on is welcome to pipe in, but I'm pretty sure, at least for this study, we didn't look at PAHs per se.
I think that's possibly one difference of the electrostatic precipitation method, where I could see if it was a filter getting covered with the soot, they'd become a pretty good trap for any gaseous PAHs, whereas if it's an electrostatic precipitator, they can flow through if they're in the gas phase. They're not necessarily going to become charged and then get trapped. So that might be one difference.
But you guys,
yeah.
Greg, just about that one thing, just because I want to make sure I'm clear about that one thing. That the particle reduction was a two-phase system. It was a HEPA filter, and then an electrostatic precipitator. So that's important.
Yeah. Okay, that is an important distinction, because then if you get a filter coated with black carbon, then it can become a way to filter gaseous components.
Yeah. In fact, we really we used the electrostatic precipitator because we weren't getting as much reduction as we wanted. I mean, I wanted to get the particles down to practically zero, which proved impossible, or virtually impossible. But the majority of the particle depletion is done by the HEPA filter.
Yeah. Yeah, I had a similar question to Victoria about the pH. So I'm glad that was raised, and also to Jeff, whether you're measuring NO2 or NOx. So whether, because I would have expected more NO coming out.
Yeah, and it might be, that one might, again, I'm glad I sort of confessed from the beginning that I'm not an aerosol chemist, but that part might be because of the way we designed our study, that it really was the balance of NO2, as Jeff hypothesized and I confirmed, but the NOx itself wasn't significantly changed.
Right. Yeah, no, and one other thing then, because you mentioned the literature. So with the available literature, would a increase of a factor of almost 4 of NO2, would these sort of effects be expected? Is that known, or is there any need to try controlled tests if it's possible with just NO2 to see if this sort of effect can be produced by just NO2?
Well, so first of all, I think it would be great to do it with just NO2 in our own hands. But the literature, and again, we didn't know that the average parts per billion of NO2 is going to end up being around 200, but that happens to be really similar to studies that were done in the 80s and 90s. And it's just like on the tip of my tongue, the investigator. But anyway, yeah, very, in retrospect, really, again, very much in line with those previous studies of NO2 alone. Yeah. Great.
I see Arthur's got a hand up, and let's see if there's any other stuff. Well, go ahead, Arthur, while I see if there's others.
Yeah. Thanks, Chris. It was an interesting talk, and I will not ask an aerosol question because that would not be fair. I'll ask about the nitrosative stress, because I think that's an hypothesis or a mechanism that we seldom think about. We often think about oxidative stress. And is what you're seeing consistent with the increased level of NO2, or is it possible that maybe there's some nitrogen in some of the other pollutants, maybe in the VOCs, that are causing this nitrosative stress?
Yeah, I don't know the chemistry well, but I don't think this has anything to do with, directly anyway, with the NOx, including NO2. First of all, the particle depletion didn't due to 3-nitrotyrosine. Excuse me. What it did to the airflow. But this is, again, Arthur, I got something in my throat. This is in the blood. Sorry, this is in the urine. This is in the urine. So like, I'm pretty confident that the nitrogen dioxide doesn't go from the airway into the urine and do things that way. So it's a cascade of phenomena. And again, I think this is mostly due to the particles, because particles, at least some of them, it's controversial, but some of them do get into the systemic circulation, and then that could ultimately be reflected in the urine. But all of what I just said is very speculative, because it's a whole bunch of steps along the way.
Are there other questions?
So I have another question that would start to tie the more cellular, molecular results to the earlier results with the clinical measures, the lung function measures. Do you think that they're are they related? That the latter part is part of what's involved in the lung function, the FEV1 changes, or are they separate mechanisms and separate potential effects?
I don't know if I totally gathered the whole question, but if I did, I definitely think that the lung function is related to the NO2. I mean, that's been shown by others. It's plausible. It makes sense on a temporal basis that would be acute. But is that, you're asking something different? Sorry.
Yeah, sorry, it's Jeff. So you observe lung function changes, and that's been seen before. And then you've observed other patterns that have changed, in terms of looking at inflammatory cells and other markers in blood and urine. So the question is whether or not the lung function changes and those other changes are related.
I see. Yeah, I think, and I don't know, because frankly, the big disadvantage of our model is that you take a 3 to 4 year period to do it, and then if there's something in retrospect you wish you'd you just can't. But and that's not an excuse. It's just we try to choose and hopefully choose the best of the options of the variable exposures. But I think what happens is that short term, as in within the first couple hours, the NO2 is highly inflammatory. There's a constriction of the airway, narrowing of the airway. We can measure it, and then everything else I am showing, I think, is independent of NO2 or largely independent of NO2, and then it becomes a matter of the particles primarily. And that's the pattern I see, and it makes sense based on what we know, because particles will stick around for hours, as I said. But I can't prove that at this point.
I guess I kind of asked that because I'm thinking about the harder part of the picture, which is how do you communicate this to someone who might want to make a decision, and whether you sort of say, well, the big effect is lung function, and then these other measures that we do kind of add more plausibility to the lung function change, or whether you would communicate the others as saying, and in addition to the lung function changes, we see all these other markers of change in the body that are preclinical changes, so that it's sort of additive in terms of the the sort of burden that your studies suggest, as opposed to sort of part of the same mechanism.
Yeah. Yeah, I do. I mean, that I'm glad you're thinking that way, because that's kind of where I'm trying to go with all of this, is what does it really mean to policy and engine technology? I think in a nutshell, it means to me that we want to get rid of traffic-related pollution in general, which is what I've always said, and anybody reasonable, I'd say, I'd be so bold as to say anyone reasonable should say, let's get rid of the source, and then whether it's the NO2 or the particles, the pollen from the trees by the highway, or major roadway, etc. All of that becomes less of an issue. But I think that it's hard to it's hard to discount that acute airflow effect, because that's the one that's the closest to clinical for me. So even though our subjects don't complain in the brief time they're with us, again, if that's happening over and over again, in the way I described, I'm worried that that has more concrete clinical consequence, because it's just that's airflow is kind of the common final pathway for lung function.
Chris, if I may plug in a bit on your comment. Actually, Chris didn't show today, but we also measured metabolites that are detectable in urine, and some of the things that we measured was urinary eicosanoids, things that are well studied in the 90s and 80s that are known to trigger the airway hyperresponsiveness and airway decrease. And what we show is that, at least in our setup, that those metabolites or those lipid mediators that are well studied to cause the airway hyperresponsiveness are elevated. And also, interesting thing was those people who had the genotype that was more prone to a more greater decrease in airflow were the ones that had a greater increase in those mediators that are shown in urine. So I think there's a strong plausibility there that that links what we measure as a biomarker and the clinical phenotype that we measured.
Nice. Okay, thanks, Min.
That's why Min's a hot commodity these days. So.
All right. Well, on that happy note, I want to thank you for a fascinating talk, Chris. You've given us a lot to think about. The policy implications are quite significant. So I really look forward to seeing how this work progresses. Because I think it'll provide some important evidence in terms of how the vehicle technologies need to evolve, or whether because we do need freight, we do need ship-ins, so we can't just say take all the trucks off the road. We've got to find a way to replace them or electrify them or do something. So thank you so much to everyone who joined us. Great to see you, Chris. Your camera's off again, but so at least you can see you to say goodbye.
Sorry, I don't know how that happened. Did that only happen at the very end?
Unfortunately not.
Oh, no, sorry.
Yes, so all the enthusiasm came through on the voice.
I
was, yeah, no, that's definitely what I had in mind. I could see the hands going in my own mind, but others might not have caught that. So thank you for a fascinating talk, and look forward to seeing how this progresses. Thanks,
guys. Really appreciate the opportunity.
Thanks for joining us, Chris.
Cheers. See you soon.
All right.
Thank you.
See everyone. We've got Vishal Verma is going to give us a talk next month on oxidative stress, so that'll fit in nicely with what Chris told us about today. So come back and join us again at the start of February.
Cool. Bye, guys.
Great.
See ya.
Bye-bye.